Yeah, so for those of you who don't know me, I'm a postdoc working with Matt Lambert-Ralph's group, and my work area is dementia. So I'm going to be talking about some transdiagnostic studies that we've been doing and how we've been using data-driven methods to understand cognitive and behavioral variation in dementia syndromes. So before I get into the crux of the whole, the data and all that, I want to take a step back and just kind of have us all orient to a fundamental question that has been driving the motivation for opting for these transdiagnostic approaches. And that is, well, what is the nature of the data that we're working with? So we know that there are two forms of data. There's categorical data and continuous data. And it's really important that we keep in mind with the kind of data that we're collecting in the clinic and, the kind of data that we're analyzing from patients to really understand what the source of this data is. Some data in the natural world is, or rather some data in our environment is naturally categorical. So like, tools that are used for hunting and cooking, they're very different. Whereas some data is naturally continuous. So, like, Matt uses an example of, growth or pressure or light. We may use some sort of shorthands such as, dim versus bright, which are kind of telling us about certain areas on the grid where we're pointing to, but that does not mean light is itself. Luminance is categorical. And similarly, once we know what kind of data we're working with, what's important for us to understand is how well do the tools that we use capture this sort of underlying source of variation. So are the tools we're using, are they actually categorized? Are they capturing some sort of binary process? So does the damage exist or does it not exist, or some sort of gradation? And are they actually capturing single or multi-process? That's what's written there. It's not visible, but single process versus multi-process. So for us in cognitive neuroscience, it's very it comes naturally to us that when we think of a certain cognitive task, it's typically many processes involved. So, memory is not just memory involves executive function, language, and so on. But many times when tools are translated into the clinic, the tool itself is thought to be an index of just that one cognitive function. And so, if someone shows a memory deficit, on a memory task, it's thought that, they actually have a memory problem, but it might actually be driven by other processes as well. And finally, is how well do the analyses that we use reflect the source? So, if it's categorical data, then group differences or trying to understand whether a certain deficit is present versus absent between two groups is more appropriate. But if you're trying to grade the severity of the deficit, we should actually be opting for some sort of a dimensional analysis. So with that in mind, how do we conceptualize this heterogeneity in the clinic? So in clinic groups, we think of, two patient groups that might be looking fundamentally different from each other, and that's what promotes this sort of between group heterogeneity, but everyone who's classified with a certain diagnostic label should be similar to each other, and that's what makes that particular diagnostic label, or rather everyone who falls under the diagnostic label, homogeneous. But this is what we predict, right? And most of the times with dementia, by the time patients come into the clinic, this is what it actually looks like. People are already on the edge of their diagnostic boundaries. They're typically showing symptoms that are characteristic of different diagnostic syndromes, independently classified diagnostic syndromes. And it's quite hard to capture these people who are kind of in the middle of that spectrum over there. And similarly, when we are using a task to measure some sort of cognitive function, one of the things that hang on, I'm just going to try to move that. What we don't realize is, that's better, I think, is that when we see a certain performance, say a person's performing bad on a memory measure, not everyone might actually have a fundamental memory deficit that's driving that sort of poor cognitive performance, but it might also be a combination of other cognitive dysfunctions. So why this is important is we need to capture this between and within group heterogeneity, but also we need to capture these common and unique factors that might be contributing to these different symptoms that we see. So how do we go about capturing this sort of sample variation that we see? Typically, in many dementia clinics, we're used to using very strict categorical sort of bins. So, like if there's an individual who presents with a behavioral problem, primarily they might go into a behavioral sort of category. Another person presenting with an attention problem might go into an attention category. And individuals who might present with a combination of these two deficits, many times it's thought that they might be actually reflecting a new subcategory of the disease or a new entity itself, But we've been moving away from this categorical binning approach to a more of a symptom-centered approach, where we are now starting to characterize these deficits happening in a transdiagnostic way, which means that we're now starting to characterize the presence or the prevalence and the severity of a certain symptom across categorically different variants. And the idea is that if you sample enough number of patients from different sort of cognitive, different kind of cognitive domains that they show deficits in, we can create some sort of a multidimensional space. The idea is that this kind of multidimensional space is able to capture these prototypical patients, those individuals who are neatly fitting the textbook definitions of certain disorders, who kind of fall in this kind of area, but also individuals who might be intermediate phenotypes or individuals who kind of show symptoms that are characteristic of two or more different clinical phenotypes. So the other thing is, how do we now, if we now have an idea of how we can capture, the intermixing of these different phenotypes at the clinical level, how do we capture changes in their performance on cognitive and clinical measures that we use? A very common approach that is used is clustering analysis. So, that's supposed to be cluster 1, 2, and 3. Sorry, but basically looking at covariance patterns within the test, and like how similar individuals are within a certain test and within across tests to kind of create some sort of clusters. But we've been moving towards using like completely dimensional approaches that where we don't, we try to not resort to a lot of clustering. And what that allows us to do is actually reduce these large test batteries that we have into some sort of fundamental underlying dimensions that might explain the majority of the variance across these different measures. The idea is that if the test itself is a good test that is used in the clinic, that is known to tap a certain cognitive function or certain cognitive dysfunction, and then you have 2 or 3 tests that are sort of being captured by one underlying dimension, we can be fairly confident that that dimension might reflect the sort of cognitive process that the tests themselves are measuring. And then we can extract what the profiles in these dimensions look like. So what's important to know is that tests do not load in a categorical manner on these dimensions. They're all relative contributions to each dimension. So, for example, in this example, you have test 1 that seems to be loading on well to this dimension, but test 2 and 3 are also loading on well, not that they don't load on the test at all. And finally, we can plot these dimensional scores that in themselves are reflective of some sort of underlying computational process in these scatter plots. And then once we project everyone's diagnostic label back, we can then see where individuals lie within this this kind of diagnostic, this dimensional space that we're trying to create. So the simplest approach that I'm going to be talking about today, and one that works really effectively, that we use quite a bit, is principal component analysis. So in simplest terms, we take all of these tests, we put them into the PCA, and the PCA derives these components that try to explain most of the variance of underlying performance on these measures. You can have as many components as many tests you have. So if you have 20 components, 20 tests, you can have up to 20 components. And in a non-ideal world, if every test is completely different from each other and no 2 tests share any sort of variance, you would have 20 components emerging. But that's typically not what happens, because tests do share variance, and performance on one test can also affect performance on other tests. And in these PCA analyses, we can then extract the number of dimensions within the space. So the first dimension is the kind of the axis that explains the majority of the variance in the data. The second one is explaining the greatest variability of what's left. And the third one is explaining the greatest variability of what's left, and so on. Dimensions can also be orthogonal to each other. So there are a number of rotations that we can perform that might allow us to extract dimensions that are uncorrelated from each other. And then essentially what this tells you is you can extract this the amount of variance that is explained by each of these dimensions. So here, what you see is, this is just an example. The first dimension, for example, explains about 21.5% of the overall variance, and this first and the second one explains about 18.5, and the first and second combined explain about 40. So because the PCA is simply a linear combination of the tools that go in, we can get components that explain. Ideally, all the tests that go in would explain all of the variance, but we're trying to explain the majority of the variance using, an ideal number of underlying components. So in dementia, how have we typically done this? We take individuals who are categorized or classified under categorically different diagnostic bins, who have undergone a lot of cognitive testing, and then we put all these patients and all these tests together into the PCA. So we're not concerned about whether a certain person is belonging to a certain diagnostic label, or we're not putting only one specific diagnostic label into the PCA, because we're trying to capture heterogeneity across the entire spectrum of performance, across the entire spectrum of different neurodegenerative syndromes. The idea is that we just throw everyone into the PCA. The PCA gives out these different dimensions, and I'll come to about how we go about explaining, extracting the number of components and so on. And then on each dimension, now we can look at where individuals with a certain diagnostic label lies. So for example, if there's a certain group that has the diagnosis of syndrome X and another group with the diagnosis of syndrome Y, we can now, within these data reduced dimensions, examine group differences if we want. And then finally, we can use these scores as covariates in neuroimaging analysis. So this would essentially tell us what kind of underlying brain region or structure or function, whatever is your choice of analysis, is explaining the kind of performance pattern across these diagnostically independent groups. So this is just an example of some work that we are doing, which is under review at the moment. And I'm going to be talking about frontotemporal dementia, which is a young onset dementia syndrome, so typically affecting people below the ages of 65. There are 3 clinical variants of frontotemporal dementia. I'm only going to be talking about 2, so I've kind of hidden the third one. So the first one is behavioral variant. So from the name behavioral variant, you can kind of infer that these are individuals who show disturbances to social conduct and behavior. They show personality changes, loss of insight, empathy, apathy, and so on. And this typically emerges from an early degradation of the prefrontal cortices, as you can see over here. And then under the frontotemporal dementia spectrum, there is another syndrome, which is called semantic dementia, who are individuals who show early sort of profound loss of conceptual knowledge. So these are individuals who sort of have a lot of difficulty, recognizing conceptual things, like naming items, because they have loss of semantic cognition. In the context of fluent speech and spared repetition. This typically happens due to early degeneration of the anterior temporal lobes. So like you can see here, this is typically left lateralized in the majority of patients. In a minority of patients, these individuals show right lateralized atrophy, but in the context of bilateral atrophy. So they have actually damage to both anterior temporal lobes, or rather degeneration of both anterior temporal lobes, but it's more lateralized to the right. So now, in the clinic, a number of patients typically present with combinations of symptoms that might look like a mixture of BVFTD and semantic dementia, right? So this is where we kind of struggle to diagnose these individuals, what kind of clinical condition do they have, and so on. And in neurodegeneration, the clinical label matters quite a bit, because because the clinical labels relate to the underlying pathology. So, they're quite strongly predictive of the nature of pathology that we suspect in these individuals. So since a number of individuals show this sort of, at the clinical level, sort of intersection of profiles between these two categorically distinct syndromes, showing a combination of behavioral, language, social cognition difficulties, the question is, are these individuals simply a different clinical variant of the disorder? Is this just happening because of disease severity? Or is this just a naturally occurring sort of a feature that pervades across the clinical spectrum of FTD that we haven't conceptualized in terms of a graded prevalence of symptomatology? So in this study, which is a collaboration with the Frontier Young Onset Dementia team in Sydney, what we did was we had 31 BVFTD patients and 31 semantic dementia patients. In the semantic dementia patients, we included individuals who have radiologically right lateralized as well as left lateralized semantic dementia. So basically just treating them as one heterogeneous cohort, really not being bothered about what clinical labels do they have. We're just putting them all into the PCA. All of them had undergone detailed cognitive assessments, indices of behavioral and psychiatric changes, as well as structural T1 weighted MRI. Essentially, we took all of these cognitive assessments, behavioral and psychiatric indices, just put it all into the PCA, and then use the dimensions that come out of the PCA to feed it into the imaging analysis. In this case, which was structural MRI, so we used VBM. So what you see here are our analysis suggested we have 8 components that seem to best explain the majority of the variance. So here in this list, I've just kind of bolded what these metrics are. So, just because these are kind of harder to read, the first few metrics are measures of general cognition, and then we have attention, working memory, and so on. So our analysis suggested that we have about 8 components that explain about 70-ish% of the overall variance in the data. And here I have some labels that just, the labels are just shorthands to refer to the component, and by no means do they explain all of the tests that load onto the components, just for ease of understanding. So what you see is all these bars that cross these dotted lines, we can think of as tests that are kind of loading highly on each of those factors, right? So here we see a number of behavioral variables. So you have, for those of you who are in the front row and can read, like it's a bit like clearer, we have measures of motivation, sleep, mood, abnormal beliefs, and so on. We have a dimension that comes up, which is semantic functions. We have general cognition, executive function, and so on. And what's important to know is that this is emerging across the entire group. So this is not necessarily coming out of only one patient group over the other, but it's actually dimensions that are coming across categorically distinct syndromes being put into the PCA together. So in the next step, what we can do is examine patterns of where each of these individuals lie by actually projecting everyone's factor score back into the dimensional space and looking at where each diagnostic label lies. So on the left plot, you see rain cloud plots. So you have these density distributions on the top, which individual points. The black line that you see is the lower bound of control performance. So anyone who is closer to the black line is closer to the lower bound of control performance, in this case, which is minus 1.96 standard error of the mean. So instantly, one of the things that you see is that for the behavioral factor, which is where behavioral disturbances are typically thought of to be very uniquely representative of behavioral variant frontotemporal dementia, that's why you have the behavioral variant in the name as well. We see quite a lot of overlap between these categorically distinct syndromes. So, there's quite a lot of overlap between individuals who have BVFTD here in the orange, as well as individuals with the right and left variations of semantic dementia here in the blue and the green. On some measures, you do see some sort of a demarcation emerging. So, for example, the semantic factor, the semantic cognition component is one of them, where you can see the semantic dementia patients kind of start pulling towards the bottom of the performance index, because this is something that is suggesting that this is something that is unique of their diagnostic sort of bin. But you see now that there's quite a lot of heterogeneity across these different FTD variations. There's quite a lot of overlap at the group level, and you have individuals within the same diagnostic group who are performing really well as well as really bad. So another way to see this is through these scatter plots. And again, individuals who are closer to these black lines are ones who are performing closer to the control standard error, control lower bound of performance. I'd like to direct you to this this last plot where we plot behavioral changes against semantic dysfunction. And it's really interesting because now you have individuals who are, better on behavioral factors. Okay, running out of time. So sorry. So just, then we can plug this back into the imaging as well. And I just want to end on this important slide, which is just some considerations that you might want to take when you're doing these kind of PCA analysis on behavioral data. First is to give real thought into the nature of the data and the research question. Are you tapping into some sort of functions or performances that are inherently categorical or continuous? An important thing is missing data, because PCA cannot handle missing data. We do quite a lot of missing data imputation with this data set. It's important to check the dynamic range of scores. So you don't need to make sure that you don't have floor or ceiling effects, but there should be some sort of variation. And obviously, two very important things is how many components to extract and what rotations to use. So since we're short on time, you can talk to me about that. And that's my last slide. Thank you.
Thanks.